So I will share my screen. Being a little slow. Everybody see that? You can unmute, say yes.
Yeah, all clear here.
Yep. Okay, cool. So I'll assume you can all see that then. Right, so as I said, my name's Rob. I'm working here at Cefas on phytoplankton. Been here about 18 months. And what I'm going to do is I'm going to talk a little bit about how and why we analyse phytoplankton. I'm talking a bit about potentially toxic phytoplankton specifically and cover some of those groups. I've got a few photos and videos from under the microscope that I'm going to show you, and maybe touch on why that's important. Unfortunately, I can't get too much into the nitty-gritty because we are short on time, but it should give you a nice little overview of what we do here. So we analyse phytoplankton at Cefas using high-powered inverted microscopes, using something called the Utemol method, which I'll touch on in a minute. For anyone who doesn't know, plankton are small, free-floating organisms in the water column, and phytoplankton refers to the plant component of the plankton. Never later, we'll talk about zooplankton, which is the animal component, and that pretty much makes up most of it. So the Utemol method involves one of these small plastic settling chambers. So we'll put a preserved water sample containing our plankton in here, and then we'll slip a glass cover slip over the top of it and allow it to settle for a certain amount of time. Then underneath here is the objective on our microscope, which will go up through the bottom and we can analyze it there. I just need to check the chat. Sorry, guys. Nevena, have you got the lobby all sorted?
Yeah, I've done it.
Okay, I'll leave that with you then. Right. So, why would you want to analyse phytoplankton cells with microscopy? They form the basis of most marine food webs. They're pretty fundamental to any marine ecosystem. So, phytoplankton provide oxygen for the environment. You may have heard statistics. I'm not sure on the percentage. I don't think people agree, but people talk about half of the world's oxygen coming from phytoplankton, and it is certainly a significant amount. Then zooplankton feed on the phytoplankton in the water, which then provide food for small fish up to large fish and so on. So if you have an understanding of what the phytoplankton are doing, that can give you inferences as to what's going on further up the food chain. You can use it as an assessment of water quality. So different groups of phytoplankton have different nutrient requirements. So if you have a lot of runoff from agriculture and things, that can change the proportions of certain groups of phytoplankton in the water. So you might be looking at things like eutrophication. Monitoring potentially toxic phytoplankton. So this is something I'm going to talk about mostly today. So if you are a shellfish farmer, you may be farming, say, mussels or oysters. Those organisms are filter feeders, and species of phytoplankton that produce toxins can accumulate in the flesh, and then toxins that wouldn't otherwise be harmful to us in the water can build up to levels such that they actually become very dangerous. And then the other thing, which is sort of closely tied with this but not the same, is harmful algal blooms. So when certain environmental conditions are met, usually temperature, sunlight, things like that, certain species can become very, very abundant very quickly. And this can lead to all sorts of problems like oxygen depletion of the water, can lead to fish kills, getting clogged in fish's gills and so on, which causes issues with fisheries and various other problems. So that's a very active area of research. People try to figure out ways to predict when HABs are going to occur. Really quite interesting stuff. But also importantly, they're quite Exhibit A. We have a few phytoplankton here. These are examples of what are called diatoms, which are a group of plankton made up of silica. So this is called a Cosconodiscus, This one here is called a gyrosigma. This lovely little sigmoidal cell here. And these are some more diatoms. These are called odontella or triaries, depending on whether the names are updated or not. And they form these lovely little chains here that you can see. So they're really quite cool. Going on to the potentially toxic species I mentioned earlier. So we've got 3 to 4 main groups we tend to look for. There are others, but around here there's sort of mainly 4. A few of these are examples of what are called armored dinoflagellates. So I mentioned diatoms earlier. If you think of the 2 main groups of phytoplankton, there are others, but there's diatoms and dinoflagellates. So this group here, containing species of what are called dinophysis, are an example of armored dinoflagellates. What all that means is that they have sort of a thick cellulose plating around the outside of the cell, as opposed to silica, which was what was in those diatoms I showed you a minute ago. So these species of dinophysis, these are all photos we've taken in our samples. These can produce toxins that can potentially lead to diuretic shellfish poisoning, which can be really quite serious. And you don't need too many of these for it to become a problem. The way you characterize them is you have these lovely little cell bodies here. So this is a Dinophysis tripos. It's got these really characteristic little legs. And the common theme between them is these structures at the top and at the side. So it looks like a little jug. These are called lists. And yeah, they're really quite interesting. These species feed on like a small cilia in the water, which is like a little protist called Mesodinium. Has someone got their microphone on? And there's some sort of feedback. Oh, thank you. If there's any questions, we will get to those at the end, I promise. But yeah, so these guys swim around feeding on Mesodinium, and they can acquire their chloroplasts from their food. And they're really, really quite pretty, as you can see, and you get sort of a variation in cell bodies. Certain species are more toxic than others. I believe this one, Dinophysis acuta, for instance, is more toxic than this one, Acuminata. Generally, the one we tend to see the most, though, is Acuminata. We do sometimes see Tripos in the autumn, which is really quite a treat, but usually it's Acuminata for us. The next one is also an armored dinoflagellate. This is a group of species called Alexandrium. These are the species responsible for causing paralytic shellfish poisoning, which can be fatal, and that can kill you really rather quickly. So it's very important we look for these. They're very tricky to identify under light microscopy. But some of the things we tend to look for are, you see this sort of Z-shaped middle bit here. This is called the girdle, and in Alexandrium they're quite deeply excavated. You can see this sort of big indentation here, and then it's offset like this. So you've got this little Z shape. So that's quite a useful feature we can use to characterize them. And then on these images in the light microscope, if you see this little line at that girdle I mentioned, that's a little membrane that runs around the girdle. So again, that's really quite a useful feature. But if you want to speciate these, you really do need to look at the cellulose plates, which is what makes them armored. And it's the pattern of those plates that will tell you which species it is specifically. A method you can use is called epifluorescence microscopy, where you apply a stain onto the cells called calcifluor. And then you look at it through a mercury burner, which produces a wavelength of light that then all those cellulose plates will illuminate, and you can see the pattern of the plates much more clearly. But yeah, these are really quite tricky and really quite potentially very, very toxic. So that's Alexandrium. And again, this is an example of an armored dinoflagellate. So all this clear area you can see, that's part of that cellulose theca we mentioned. Next one is we're going back to a diatom. or a group of diatoms called Pseudonychia. And these can be very, very tricky to spot if you've got a busy sample, particularly in the summer where the phytoplankton are more abundant. So we split these into species groups. You can't really speciate them unless you're looking through a very high-powered microscope, like an electron microscope. But the species groupings have some sort of commonalities. So this first one, Pseudonychia delicatissima, you've got very thin, straight cells. And the way to tell if it's Pseudonychia is the way they join up at the ends here. You see how these sort of two dark lines sort of overlap and sit next to each other? So that's one of the ways you know it's Pseudonychia. Also the fact they're sort of pointed and they have these sort of two chloroplasts fairly centrally. So yeah, delicatissima, straight, thin, multistrata are very characteristic. They have this lovely sigmoidal shape to them, so they sort of flick off at the ends. And then seriata is a little bit larger, so you can sort of see the cell detail a lot more clearly here. This chain is actually laying the other way up, so you can't see this overlap like you can on delicatissima, which makes it very tricky. And I've got a bit of video later. Unfortunately, it's only very, very short, but you'll see there's a chain of seriata in the video, and then some other thin chains of diatoms, which you could easily mistake for Pseudonychia. So you have to be rather careful when you're looking for these. Now, these produce toxins that can lead to something called amnesic shellfish poisoning, which in serious cases can lead to memory loss and various other symptoms. And they're really quite common. But luckily, you need quite high numbers of cells for it to pose a problem. So those are sort of some of the main toxic groups that we look for when we're analysing phytoplankton. I've got a short little video now of some sort of live cells and some of that fluorescence I talked about earlier. This may be a bit jumpy at first, but you'll have to bear with. So this, for example, is a species called Gymnodinium. This is from a culture we were given from some colleagues in Weymouth. And you can just about make out in the middle of the cell, you can see that girdle, that sort of dark line. And then this is another species called Porocentrum micans. So this isn't toxic, but it's just a very pretty sort of leaf-shaped cell. These are really quite common. We see a lot of them in UK waters. They've got sort of a characteristic spine on the end. Very hard to keep them in focus when they're swimming around like this. But yeah, these are really quite nice, and we see a few of those. I've got a fluorescent image of that later in the video. So this is, I believe, an Alexandrium, so under the fluorescence that I mentioned earlier. And you can see how the lines of that girdle and the plates are much, much clearer. The video doesn't pick it up nearly as well as you'd see it under the microscope. So you have to forgive me if the plates aren't totally obvious. But this is a different species, but you can see really clearly here those armoured plates on the theca. And again, this is a Tripos lineatum, so these would be quite easy to identify based just on their shape. But again, you can see here on the base, which is called the hypotheca, you've got sort of divisions where all the plates show, and that makes it much easier for us to speciate what phytoplankton there are in the water. This is that Pseudonychia chain I mentioned, the seriata type, and you can see here these thin chains of other diatoms could easily catch you out. And then there's another little Porocentrum micans there, just because they're quite pretty. Yeah. Now, I mentioned the Dinophysis species that can cause diuretic shellfish poisoning. This is another species that produces similar toxins and can lead to the same condition called Porocentrum lima. So we've got 3 cells here. And the way you characterize these is by these little indentations in one end. And there's very, very small plates. And with a good enough microscope, you can see those. They have a central pore in the middle, which you may be able to see on your screen. It's not the best quality on the video. But another good way to speciate these is they have a pore pattern. So I don't know if you can make these out on the video, but there are very, very small little light spots all around the edge. So you've got these, and then you have much smaller ones going in towards the center of the cell. And it's from the pattern of those pores that you can then identify whether or not it's the species you're looking at, which is particularly useful if it could be toxic. These phytoplankton can really be quite seasonal. And I've got a plot here, which is, can you see at the bottom there? I've just realised my little bar is in the way. But over the winter, so these are the winter months, you've got very, very low counts going through the winter, and then suddenly as the weather starts to pick up and it gets a bit warmer, things really do, excuse me, things really do start to spike, and you get some quite high abundance, and then it peters off again as the weather gets colder. Different species will proliferate at slightly different times. Generally speaking, it's summer. That tends to be a very busy time for us. And this is so uniform that you can actually look at these patterns over the course of years. So all of these peaks are over the summer months. And you can see this is an example of a bloom forming species. So you've got abundance going up here on the left, and you get some really, really high spikes. Now in the winter months, we are actually still detecting this species in our samples, but the numbers are so low comparatively that you just can't see it on the plot. But yeah, that seasonality is really quite strong for a lot of species of phytoplankton. Next up, and lastly, I've just got another animation for you. So I'll briefly talk about what this is. So this is a map of the UK, obviously, and any blue dots you see flashing up around the map are where we've detected species of interest below a certain level. And then and this is going through the course of 1 year. So it'll start off in January and you'll see lots of little blue dots. And then as we get into summer, you'll see other colours flashing up, which are species of interest above a certain level. And the larger the coloured dots become, the higher the abundance of those species. Particularly down in Cornwall, you'll see a lot more of this. Although unfortunately, I don't know if my little window is in the way. I don't know how to get rid of that. But yeah, you can see here, things really starting to pick up. This is sort of midway through the year in the summer months. And then I'll play this again in a minute. And you'll see it start to die down, mostly black to blues, as the winter months come around. So you can see we're getting things all around the coast of the UK, particularly in Cornwall. That's partly due to sampling, but it's also down to various hydrographic and oceanographic features, I would imagine. But yeah, so we see them sort of all year round, which can be really quite nice. And then if you're monitoring over a long period of time, you can make these really nice little time series and see how things are changing. But yeah, right, I believe that is about it from me. So I'll stop sharing now. Okay, how long was that? I think we're all good. Right, over to Nevenet. You all good to go?
Yeah, I'll just load mine up. Right, can you all see that okay? I hope so.
Yeah.
So basically, Nevenet Almeida, zooplankton taxonomist at Cefas, and I'll talk a bit about the zooplankton community, which covers the generally heterotrophic protist and metazoan component of the plankton community. So in other words, basically the animal side of things. So like phytoplankton, they cover distances in the water column by drifting as opposed to swimming. As they feed mainly on the phytoplankton, they're also concentrated within the upper 200 meters of the water column, although most of them undergo daily patterns of vertical movement known as DL vertical migration. This is typically a single daily ascent, usually on either a nocturnal or twilight basis. This makes zooplankton especially significant in mediating fluxes within nutrient cycles such as carbon. So there are several groups which zooplankton can be divided into. The crustacean zooplankton are the most dominant group, while some taxa are collectively termed as gelatinous zooplankton and include groups with extremely high levels of water and salt content in their bodies, such as jellyfish, ctenophores, and salps. Zooplankton are further divided into either meroplankton or holoplankton, the latter of which remain in the water column for their entire life cycle, whereas meroplankton, by contrast, consist of the planktonic larvae of many aquatic organisms, which are otherwise endobenthic or epibenthic. These include the larvae and egg of many fish species, which are collectively known as ichthyoplankton. So in this talk, I will just cover some examples of each of these groups, which we often encounter here at the Plankton Lab at Cefas, along with some key facts and features about each taxa. So first I will cover the crustacean zooplankton group known as copepods. These are an example of a taxa which counts as holoplankton, as they spend their entire life cycle in the water column. The calanoida order in particular is the most abundant and diverse pelagic copepod order, and these are the plankton which most frequently populate our samples, with the adults generally being larger than the adult individuals of the other main orders. So as a result, many commercial fish species are reliant on calanoid copepods within their diet. Calanoids themselves generally require phytoplankton as food, whilst other copepod orders may also be omnivorous or will also feed on bacteria. Right, so up next I've got a video. I will apologise now if they act a bit jumpy. They can not run smoothly at first. Right, so in this video, if it's hopefully playing for you, footage has been taken from the microscope, and most of the copepods displayed here are calanoida copepods, particularly from the Calanus genus. So here you can see the trusty pokey stick being used to manipulate the specimens, as we would normally do for identification purposes. So the key features we typically examine during this are such things as the number of segments on certain sections of the body, and even the number of processes or setae on individual limbs. Calanoids have long antennules, as long as the body itself or even longer. The adults are also generally easy to identify to genus level. And morphological features can also be used to distinguish between male, female, and juvenile specimens. So here we have another type of crustacean holoplankton called euphorsids, which are commonly known as krill. These are a small group of shrimp-like plankton, of which some species occur in the greatest concentrations ever found for pelagic crustaceans. Therefore, many species are of considerable importance as food for fish and some marine mammals. Euphorsids also notably possess 10 prominent eye-like photophores, which are unique among crustacean zooplankton and emit brilliant blue-green lights. So hopefully you can just about see the photophores in this footage here as the spots displayed along the length of their body. The function of these photophores may relate to mating, social interaction or orientation, and as a form of counter-illumination camouflage, which is used to compensate their shadow against overhead light. Euphorsids may additionally be distinguished by their anterior tinges of reddish pigment. They swim using their large legs, which are known as pleopods, and prey on other zooplankton by filtering them, as if with a feeding basket. So to move on to an example of a meroplankton taxa, the plankton displayed here are from the family Nephropidae, the true or clawed lobster family. This particular species is Nephrops norvegicus, which are commonly known as the Norwegian lobster and may typically be enjoyed as scampi. In fact, they have even been known as the most important commercial crustacean in Europe. So being an example of meroplankton, only the larval stage is planktonic and lives within the water column. Therefore, the highest numbers of this species are found in the north of the European region, as its distribution is determined by the availability of suitable substrate for the adults to burrow into. Right, so here the larval stage of nephrops, which is known as a zoea, is very distinctive with extremely long lateral spines. So as can be seen in this video, the pelagic larval stage is very distinctive from the adult form, which has the more typical body shape of a lobster. So taxonomists wishing to identify the developmental stage of nephrop will use the orientation and length of these spines to determine between the different stages of zoea, which in the case of nephrops is between 1 to 3. So we move on to ctenophores, which are a type of gelatinous zooplankton and are differentiated from cnidarian jellyfish with their lack of nematocysts or stinging cells. They are commonly spherical or bell-shaped. You may have often seen the spherical species stranded at the sea edge here in the UK, which are commonly known as sea gooseberries. They are characterized by cells named cilia, which form rows like combs, giving them the term comb jellies. The rows of cilia B, kind of like Mexican waves, to propel the ctenophore along, also diffracting light as they do so. And this gives off a beautiful rainbow-coloured iridescence, which you can hopefully somewhat see in the following clip. And hopefully that's playing all right. Okay, so finally we get on to the ichthyoplankton group, which is composed of the larvae and eggs of many teleos fish species. So these fish often produce huge amounts of eggs, up to hundreds of millions per female. The transparent pelagic eggs which are produced usually have a diameter between 0.7 to 1.5 millimeters, though in rare cases reaching up to 2.6 millimeters even. Oops, sorry. Accidentally went to the next slide. So they are released into the water column after fertilization and may drift between the surface and the bottom during their incubation period. The planktonic larvae eventually hatch from the eggs and will go on to feed on phytoplankton and predate other zooplankton until they develop into the adult fish. The mortality of the larvae, however, is very high, potentially occurring from inadequate food sources, temperature, offshore transport and predation. And as the survival and distribution of fish larvae and eggs has a big impact on fisheries recruitment, Cefas has been involved over the years in many surveys which aim to determine the abundance of ichthyoplankton around the UK. Right, so now we move on to this video, which hopefully, as you can see, some fish eggs can look quite distinctive and beautiful. So these eggs shown here come from an offshore species of fish commonly known as pearlside, whose eggs also rather live up to their name with their somewhat iridescent and patterned shell. But a lot of fish eggs can look very similar, however, and it's no easy task to be able to determine the exact species for every fish egg we come across. Sometimes this can be achieved, however, by measuring the egg diameter and oil globule size, if present, and paying attention to the pigmentation of the yolk and even of the developing fetus. So in this clip, some of these eggs were identified from species such as mackerel and dragonette, which I think are these ones in particular. So similar principles are applied when identifying fish larval specimens. As a fish larva grows, the pigmentation becomes more distinct. The larva may also start to develop certain features once they reach a certain size, such as spines and developing fin rays. So here in this clip, larvae have been included from species such as mackerel, megrim, and dragonettes. It does take time to develop the skill to discern these features reliably, though. Some fish species such as sprat, sardine, and herring can look identical and literally only be distinguished by counting the number of muscle bands along the length of their body. Similarly, it is no small feat to be able to reliably identify other zooplankton groups in general. So, having covered but only a small part of the entire zooplankton community. Hopefully this talk helps to illustrate the enormous diversity present within the plankton community, and therefore the varied ways they may interact with the rest of the aquatic ecosystem, and why they are basically so important, being at the base of the food web as they are for monitoring purposes. So that's everything from me otherwise, and I shall therefore now pass over to the Benthic team, Matt and Peter, who will go on to cover the benthic component of these ecosystems. Okay, so if I just stop sharing.
Sorry, I didn't quite catch that. Could you please?
All right, hopefully you're good to go now.
Hello everybody, I'm Matthew Curtis, a senior benthic ecologist here at Cefas. Just to say we are overrunning a little bit in this session. I think the main session has started again, so if you want to leave, please feel free. If not, we'll just run our last little bit quite quickly. Right, let me just share my screen. Oops. Hopefully you can see that. Right, so yes, we the Benthic Lab, we look at the macrofauna. It's anything on the seabed greater than 1 millimetre usually. If it's estuarine, we do go down to 0.5 mil. Anything less than that we consider as to be myofauna. So our samples tend to come in collected at sea using grabs, either on the Cefas Endeavour or off other vessels of opportunity, so we can get samples of multiple sizes. I'll just play a quick video for you. These may be a bit jumpy as they go, but this is just a quick video of our processing lab with the fume covered in. And yeah, as I mentioned before, samples can come from sea in various sizes, 10 litre bucket down to 0.25 of a litre, depending on the sediment we're sampling. Soft muds, we don't tend to get a lot left. Of course, gravels, and in this case, shelly sands, we end up with quite a lot of sample left. To process a sample, we are just really just extracting everything out of it. So we use this over multiple sieves. And keep retaining everything over 1 mil. So this is just getting rid of the excess fluid, formaldehyde, and keeping everything that's greater than 1 mil. And then the remaining sediment is sieved through different meshes just to make the efficiency of the sampling greater. If we just put everything on a tray or under a microscope at this size, you get you miss a lot of things. So we fractionate it first just to make our processing easier. And this is just an example of what will be left on a 4 mil sieve, a 2 mil sieve, and a 1 mil sieve from this sample. And then the floated extracts, we use water to extract the lighter stuff, and this is what would be left over for the lighter fraction. And this all gets sorted under a microscope. Yeah, and this is an example of just some of the things under a microscope, but I've actually got a live video that I'll show you in a minute. So this is just the greater fractions that we get sorted on trays just under an illuminated magnifier. So we pick through these trays, pick out all the fauna, then bring it into the shared microscope space at Cefas. This is shared with the phytoplankton and the zooplankton people you've already seen earlier. That's a video that doesn't seem to be playing at all. Yeah, so we identify all our animals down to species, and we just either using all the literature we have, and we also have quite an extensive reference collection. So if I now sharing this and go to my live video. I'm not sure if that can be seen at the moment. Let me try again. But hopefully that can be seen. So a live view camera down my microscope in the lab. So this is a sample we've got from an MCZ, a marine conservation zone, just off the coast of East Anglia, Orford Inshore MCZ. So this is just some fauna that we've picked out, and we'll then start the identification for. This is quite a coarse sample. Samples. We've got a lot of tube. Sorry, I thought I disappeared for a second. I got an on-hold message on my screen. So yep. Also under here are some Sabellaria, which is a reef, potentially a reef-forming worm. They make sand tubes that adhere to hard substrates, but they also then on softer substrates build up on top of each other to reefs that can be up to 1 metre high and quite extensive. And then a couple of examples of some other fauna we have. To try and find these on my screen. So that's a little Gammarid amphipod upside down. So on these we have to identify these, we are looking at some very small features. So we have a high-powered microscope to actually zoom in on some of these features to help in our identification. And there's just a couple of examples of some other polychaete worms, if I can get this to focus. That's a ceratulid, some description, and some are on here. Should be another little worm, a Schistomaringus or something of the like. So that's just a very brief overview. This data, we all our fauna from each sample are identified and enumerated, and that then data is compiled together and then is used for, could be characterizations of MCZ or monitoring of dredge disposal sites. I will then now pass you on to my colleague, Dr. Peter Barry, who is going to talk to you about invasive species.
Share content. Hello, hopefully everyone can hear me. I'm going to talk to you about invasive species, and particularly those that come across on marine litter. Here's a sample of a bryozoan colony, which I found on a piece of floating plastic. If I move the camera, this is a live image, and then I zoom out a little bit, you can see that this is actually just a torn off scrap of plastic, which bryozoan larvae have found. The thing is, bryozoans, they like to adhere to a hard substrate. They usually find something on the benthos, a rock, but increasingly they're beginning to find floating plastic. They find a firm substrate and they think, well, this will do, this is good enough, and they adhere to that. The problem is that it's mobile. These plastic fragments are floating around the oceans. They're just at the whim of the ocean currents. So species adhere to these pieces of plastic, and you never know where they're going to wash up. It's an increasing problem. We've provided artificial rafts for invasive species, which traditionally have been, contained in their own environment. Now they can move anywhere. So it's a problem we're becoming aware of. There was a piece in The Guardian yesterday. We've got lots more litter. I was sorting through some samples last week. I found bryozoans, mollusks, crustaceans, particularly barnacles, but also mobile fauna. So that it's just something to be aware of. I know we said we were going to do benthic, but this is also pelagic fauna. So that's it. That's my bit right at the end. Just wanted to draw your attention to that. Thank you very much. Any questions?
Okay, thanks, Pete. That was really good. Has anyone got any questions? We have overran quite a bit, but we're all happy to stay for a few more minutes. Anyone who hasn't seen the chat, the panel event started again at 12. So if you are after that, then you want to join by the link in your email. But we're going to hang around for a few more minutes if you have any questions. You can either unmute or raise your hand via the options at the top. It's up to you.
All good then.
I'll take that as a no. All right then. Well, thanks everyone for coming, and yeah, enjoy the rest of your day. We have an afternoon session again later on, I believe. Do you guys remember what time it is? Is it 2 o'clock?
2:10, I believe.
I think it's just people here now, but all right. Okay, so I'll save that recording and we'll go again this